everyone. Let's start from where we left in the previous class, where we were started to discuss how GPCR signal. We discussed different properties which are associated with receptor. We also saw different types of G alpha which are required to take signaling forward in different direction. We will now talk in today's class, or we will now discuss in today's class, how G protein coupled receptors respond, what kind of signaling they perpetuate, and so on. Before we go into details of that, let's discuss some new terminologies, and to some of them I introduced in the previous class. Here in case of GPCR, what are the new terminologies which we will be seeing? So I already told you that often ligand is called agonist, and the idea of agonist is that it should bind to the receptor and activate it, then only it is agonist. The activation could be a complete or a partial one. So agonist could be a full agonist or it could be a partial agonist. Then there has to be an inhibitory type of molecule which is called antagonist, which will bind and compete with, it will compete with the agonist and will bind to receptor. And the job of antagonist will be to bind and let agonist remain free from the receptor and not activate the signaling. So it will bind but it will not activate the signaling. Basically it will inhibit the receptor functions. Then we saw in previous class some of the terminologies like RGS I told you. There is another molecule which is new to this GPCR signaling, or which we have not seen in earlier types of signaling, is called guanine nucleotide dissociation inhibitor. Basically it will ensure that G alpha remains in GDP bound state or in off state. Then we have already seen GEF, which was already known in many cases which we have seen earlier. However, uniqueness was that this GEF is associated with the receptor itself. We discussed RGS, which was equivalent to the gap activity, where G alpha T was being hydrolyzed into G alpha D. What basically it means that these gaps and GEFs, normally also these process happens, but their rate is very poor. So with the help of GEF, more GTP molecule can be loaded in given time onto G alpha protein. Similarly, there is basal hydrolysis of GTP which can be done by the G alpha, but this is accelerated by multifold when gap is provided. Then we will see some of these molecules which are called GRK. GRK, basically G protein regulatory kinases. When we will see that signaling has happened, now the response to shut down, then you will see GRK kind of proteins coming, and they will basically bind to the C-terminal tail region here and phosphorylate certain residues. This phosphorylation is done by this GRKs, which are example of serine threonine kinase. Now let's see some more interesting aspects of GPCR. What exactly happens over there? So let's take example of simply a receptor. I told you that GEF activity is part of the receptor itself. So there is always some basal activity which is associated with the receptor. However, the residence time of the G alpha in ligand unbound form of receptor is poor. So the exchange is very poor. However, some receptors could be there where G alpha spends more time with the receptor even when it is not GTP bound. So there will be some receptors which will be active to a basal level. It's not like that inactive molecule is moving to the active molecule. Rather, there is an intermediate situation where receptor has basal activity, and from there it can be moved to the complete activity or full activity. So if I say the receptor as R and activated receptor as R star. So your agonist will ensure that your R goes to R star, and in presence of antagonist, basically if you compete out agonist with antagonist, it will be taking back to the inactive state of the receptor. Then there is this intermediate state which will be there, and from here the receptor can go to the complete active state or inactive state. We all understand that there will be some different molecule, agonist type only, which will be taking them into different configuration. So agonist and antagonist play very important role in this way. So as you would understand, the R going to R prime, this forward signaling or forward movement of the reaction will be done by agonist. R prime or star activated goes to R. If I say in inactive receptor, this will be done by antagonist. Also, there is something called inverse agonist in this case. Basically, inverse agonist means that this state of R, which was the intermediate stage, from here the receptor will go into the inhibited state. So it is doing the job which is opposite to the antagonist. That is why this is called inverse agonist activity. Remember, some of these inverse agonist activity will be shown by those receptors which have basal activity in them. Not all GPCR will have basal activity, so the ones which have basal activity will take this intermediate state to inactive state by the availability or binding of the inverse agonist. The agonist which is here not only will be able to take R to R star, but also R intermediate will go to R star. So this is also done by agonist. And if you think about the antagonist, we have already seen that how it will ensure that activated receptor here goes to inactive receptor. Now the other thing which basically will happen here is that there is something called full agonist, which I told you. Sometimes this is also called pure agonist, full agonist or pure agonist. As I said, this will be taking the receptor into active state. And what inverse agonist will do? It will take the intermediate into the R activity. So basically the ligand or which is agonist is coming in different flavors. It could be full agonist or pure agonist, it could be a partial agonist, or there could be something which is similar to agonist, which is antagonist, and a variant of antagonist or similar looking molecule is inverse agonist. Two of them are taking signaling or the reaction in the forward direction, which are full agonist and partial agonist, and the ones which are taking in opposite direction are called inverse agonist and antagonist. Something of this sort we did not see in other signaling. There only thing which we noticed was that ligand is available or ligand is not available. Ligand is a monomeric version, it needs dimerization to be active, but we did not see these inverse or antagonist activities which were there. Antagonist only in terms of that you will find an inhibitor to which the ligand will be bound. Here antagonist does not bind to the agonist, rather it binds to the receptor and competes for the agonist binding. Now if we talk about the GPCR and the different agonists and try to look for the signal, what are the responses they mount? If you consider this as the agonist concentration, suppose I say micromolar, and here we are checking the response. Response in the sense that suppose if the receptor was supposed to activate and generate cyclic AMP. So how much of cyclic AMP is forming, that basically is the response. And let's look from the perspective that there are receptors which have this basal inherent activity. So what will full agonist do? Often you see this response. So this is your full or pure agonist. What will partial agonist do? It will activate to a different level. So partial agonist could be entirely different molecule, or it could be derivative of the full agonist. Depending on what type of partial agonist it is, how much different it is from the full agonist, you will see the variation in this response which is mediated by partial agonist. If you think about the antagonist, so this will ensure that this basal activity is there and this activity is inhibited, not that it is activating the receptor and the kinase, or it is not the receptor which is getting activated, rather it is ensuring that no activity moves out of the receptor. So your antagonist is ensuring that this basal activity level, or if there was no activity, it comes back to that level. What inverse agonist does is that this basal activity is brought down further below. So this will be done by inverse agonist. Remember, whenever we are talking about inverse agonist, which we may not discuss much in coming classes related to the GPCR signaling, but wherever this concept of inverse agonist come, you are thinking about some basal activity below which this inverse agonist is driving the reaction. So if this is a positive response, this is basically the negative response. Now if we look about simple GPCR family, the receptors itself, there are a huge number. We will see more and more detail how typically these components come together. So these receptors can be classified into 6 different classes. In the beginning I told that there are 6 classes of receptor, and I also told you depending on what ligand they were binding, what kind of activity they were mediating, what process they were engaged in, accordingly they have been named. So class A is called rhodopsin type. Basically here one of the first molecule which was studied was the pigment which is in the eye and rhodopsin, through which we had the visual perception. Then there is something called secretin type. Then you have glutamate receptors which actually bind to glutamate and respond to that. Then there are some GPCRs which are first characterized in the fungus, where they say MAT alpha, MAT. In case of yeast we know that we have MAT alpha and MAT A kind of conditions with the yeast. So over there this pheromone type mating situation is being controlled by GPCR, and that is your class D. Then there is something called cyclic AMP receptor like. So there are molecules which will be binding, there are these GPCRs which will bind to cyclic AMP and then do the signaling. Not only that, it is involved in the cyclic AMP generation or AMP generation, but also it is important by responding to this AMP like molecule. Then the last class is called frizzle like or wind signaling. These are the molecules which play a very important role in developmental process. But largely you will see that GPCRs are responding to small molecules, hormones and biological amines like neurotransmitters and so on. We have already seen that There are almost 800 genes which code for GPCRs, but almost half of them are involved in sensory perception of smell, taste and vision. And a very good number is still an orphan receptor. So the other type of receptors which are engaging in activities of this sort, they are very less in number. So basically they respond to small peptides, large proteins, hormones and neurotransmitters. Now if you think about the signaling moving forward, so downstream of the receptor, you will need G-alpha recruitment. I told you this G-alpha is of 4 different types. One of them is called G-alpha-S, I already told you. Then next one is G-alpha-I, then there is a G-alpha-Q, and then you have G-alpha-12, 13. What we know that they engage in cyclic AMP production. This reduces the cyclic AMP or GMP level. So these use enzymes which are in the class of cyclase, and these use the enzymes which are in the class of phosphodiesterase. This one works through PLC beta, and we know what PLC beta does. It breaks down PIP2 into DAG and IP3. These ones directly talk to small GTPases of Rho, RAC and CDC42, and they together regulate cytoskeleton. So 4 major classes of G-alpha, ensuring that signaling goes in different direction. We will see some example of each one of them as we go along in this class. So basically G-alpha-S can be considered as the first class, second class is G-alpha-I, third is G-alpha-Q, and fourth one is G-alpha-12, 13. Here we have seen multiple other combinations also, where olfactory, many of the olfactory sensation, meaning what we smell around us to different ligands, is mediated by G-alpha-S. So some of these G-alpha can also, you can also see that some of these G-alpha-S are also named as G-alpha-olf, because they are involved in the olfactory sensation. Similarly, G-alpha-I, we know that it will reduce the level of cyclic nucleotide. There are examples of G-alpha which is labeled as T for transducin. See, one of the G-alpha which was explained in case of visual perception in rod cells, they work through by reducing the cyclic nucleotide level, and that is why they are in the broader class of G-alpha-I, but just for the sake that it was identified with the visual perception along with this molecule which is transducin, this is called G-alpha-T. Here we know that this will, so here we are talking about only the G-alpha, and I told earlier that sometimes G-beta gamma also participate in signaling. So when it comes to PIP2 mediated DAG generation and IP3 generation, this helps in calcium release, and this helps in activation of PKC, protein kinase C. So even G-alpha-Q along with the even G-alpha-Q and G-beta gamma of its heterotrimeric combination, they work on the PKC activation. Then G-alpha 12-13, they have mostly the G-alpha type which is getting activated, and they work through rho RAG GTPases, as I said. So they promote the rho GTP bound state. So they will work through the rho JEP molecule. Now let's see a simple example of the signaling by G-alpha types, and particularly the G-alpha cyclic AMP generation. So in coming slides we will see particularly the action of Galpha-S, where it will use the ATP molecule and synthesize cyclic AMP molecule. Cyclic AMP generally works through the protein which is called protein kinase A. So if we understand this particular response, and as I said, many of the G-alpha-S are also called G-alpha-olf. So now think about a receptor which is there in the membrane, and it recognizes, binds with a smell molecule, any aroma, and this it will bring the G-alpha-S, activate it, and this activation will look for the molecule which is also membrane bound, and this enzyme is called adenylyl cyclase. Because it is helping cyclization of adenyl residue from ATP to cyclic AMP. So the binding of ligand to the receptor activates G-alpha-S, which activates the adenylyl cyclase, which helps generate the cyclic AMP. And this cyclic AMP will work through protein kinase which is called protein kinase A. Let's see what happens in the case of protein kinase A signaling. So once cyclic AMP is generated, protein kinase A molecule is a heterotetrameric assembly. There are catalytic unit. If I say 2 catalytic unit and 2 regulatory units. So normally in absence of cyclic AMP, this heterotetrameric assembly will be there, and the catalytic activity of this unit is regulated by this regulatory unit. So what cyclic AMP will do? It will go and bind to the regulatory unit. Once this binding has happened, what will happen? That there will be dissociation. So catalytic units will be separated from cyclic AMP bound regulatory unit. This again emphasizes on the fact which I told earlier, that in case of signaling you will see that small modification which happen to signal, receptor, effector, adapters and so on, that changes the affinity for certain proteins and protein-protein interactions. So here the unavailability of cyclic AMP and this regulatory unit free was binding to the catalytic unit and keeping it inactive. As soon as the cyclic AMP bound, the affinity for the catalytic unit decreased, and that is where your catalytic unit is free to do what job? Basically this is a protein kinase of serine threonine kinase type. Okay, so this will now find its molecule, target molecules and phosphorylate. This is where amplification of the signaling further goes on, because cyclic AMP which is generated, that will activate hundreds of pKa molecules, and each pKa molecule can phosphorylate the target, hundreds of target molecules. So target protein is phosphorylated. And one of those target proteins can travel inside the nucleus, where it will bind. So, this pKa molecule, in addition to phosphorylating targets, it can travel inside the nucleus also, and this active pKa will phosphorylate molecules which are present over there. So, there in the DNA there is a sequence which is called cyclic AMP response element, to which there is a molecule called CREB is bound. But this binding can happen only when pKa is active pKa is inside the nucleus, and that has phosphorylated the CREB. So, you will get a phosphorylation tag. Now, this CREB, which is phosphorylated, binds to this region, and through help of other protein, that will activate the transcription process. So, you notice that how a small molecule binding to the receptor at the cell surface activated the G-alpha, which activated a enzyme which is cyclase family of enzyme, and that enzyme actually ended up activating the molecule which is pKa. This pKa moves into the nucleus, phosphorylates this protein which is called cyclic AMP response element binding protein, CREB, and this phosphorylated version of CREB binds to the elements where transcription of target genes is regulated. So, this is one of the G-alphas downstream of one class of GPCR, which is ensuring that signaling goes on in right direction with the help of pKa activity. So, now if we try to see what we have understood in this class today, that GPCRs is the largest family, we have already known this, but this is present in almost 6 families, and out of which multiple of them, almost half of these GPCRs are responding to the sensory stimulus which is present around us. Then the G protein as such, which is working downstream of GPCR, there are 4 main different types: G-alpha-S, G-alpha-I, G-alpha-Q and G-alpha-12-13. GPCR can actually exist in different conformations, one which is active, inactive or intermediate state, and this receptor bound active G protein will activate the effector protein for signaling to continue. We saw example of adenylyl cyclase as the effector molecule, which was generating CAMP, which activated pKa, and this pKa which is generated through cyclic AMP will be mediating the response inside the nucleus by affecting the transcription through CREB-like molecule. So, what we understand that GPCRs are playing very important role. There are multiple different types and each one of them is giving rise to a specific response. Thank you.